Welcome to today's Clinical Microbiology session. In this session, we will discuss about TSI test, an important biochemical test used to differentiate enteric bacteria. Triple sugar iron agar test. This is one of the most important biochemical tests used for identification of gram-negative enteric bacteria. So, the media used is triple sugar iron agar. It is a composite media. Composite media means it is a type of media that can demonstrate different properties of a bacterium. Examples: TSI provides three different properties of a bacterium. First, fermentation of the sugars in the form of glucose, lactose and sucrose. Then gas production in the form of carbon dioxide and hydrogen gas. Then hydrogen sulfide production, that is H2S production. Components of TSI agar. So three sugars are incorporated in the media, that is glucose, lactose and sucrose. Sodium thiosulfate and ferric ammonium citrate are important substrate for H2S indicator. Phenol red acts as the pH indicator. What is the purpose of TSI test? Used to differentiate the members of Enterobacteriaceae family from other gram-negative rods. Second, to know the ability of bacteria to ferment glucose, lactose or sucrose and forms H2S. Methods of preparation. Concentration of TSI sugars like glucose, lactose and sucrose in the concentration of 1:10:10. That means glucose is 1 part and 10 parts are lactose and sucrose. The media solution is allowed to solidify in the slant. This configuration results in formation of two reactions in the same tube. The slant is mainly exposed to oxygen, so it creates aerobic condition, and but it is not exposed to oxygen, so it creates anaerobic condition. Keep both slant and butt in equal length, approximately 3 cm. Why slant and butt are made in equal length? To preserve both aerobic and anaerobic chamber in the test tube. Proper inoculation of TSI media. So it is mainly inoculated by two methods, first tub method and second streak method. So first tub the butt with straight wire loop, then streak the slant in zigzag manner. Never touch the bottom of the test tube with straight wire loop. Incubate at 37 degrees centigrade for 18 to 24 hours, then interpret the result. The principle behind the TSI test. A glucose fermenting organism turns the entire medium acidic in first 8 to 12 hours of incubation. So please remember the incubation time, that is 8 to 12 hours of incubation. Then in slant person glucose is oxidized, but in butt person glucose is utilized anaerobically. As a result, the entire slant and butt remains acidic or yellow. After that, the glucose supply is exhausted and bacteria begins oxidative decarboxylation of the peptones within the slant person where oxygen is present. The amino acid produced by degradation of the peptones is insufficient to change the acidic environment of the butt person. So the reaction appears as K by A, that is alkaline slant with acidic butt. How H2S is produced? Sodium thiosulfate, which is present in the media, mainly form hydrogen sulfide, but it is colorless. It reacts with the ferric ammonium citrate, which is also present in the media, and form ferrous sulfide, which mainly appears as black precipitate and responsible for blackening of the medium. Always remember those key points for identification. First, yellow medium indicates acidic medium, then red alkaline medium, bubbles in the medium or complete displacement of the medium from bottom of the tube or slight indentation of the medium indicates gas production, blackening of the butt H2S production. Results should be interpreted at 18 to 24 hours of incubation. Next, interpretation of result. Remember, always read slant reaction first followed by butt reaction. So K by K reaction means alkaline slant and alkaline butt. It indicates that no carbohydrate fermentation, bacteria is non-fermentative. Example is Pseudomonas aeruginosa. Next, K by A reaction indicates glucose is fermented, lactose or sucrose not fermented. characteristic of non-lactose fermenting bacteria such as Shigella species. K by A with H2S, characteristic of non-lactose fermenting H2S producing bacteria such as Salmonella, Cytobacter species and Proteus species. A by A with gas, glucose or lactose fermented, characteristic of lactose fermenting coliform bacteria. Examples are Escherichia coli, Klebsiella species, Enterobacter species. Viva questions: why is the slant red and but yellow in K by A reaction? Glucose is present in low concentration, that is 0.1%. After glucose is exhausted, the organism utilizes peptones aerobically on the slant, producing alkaline products, while the anaerobic but remains acidic due to glucose fermentation. Let us discuss the TSI result and its interpretation. A by A with gas, this mainly interpret as glucose plus lactose or sugar fermentation. Examples are Escherichia coli, Klebsiella pneumoniae. K by A reaction mainly indicates glucose fermentation only. Examples Shigella species. K by A with H2S production, glucose fermentation plus H2S production. Example is Salmonella species. K by K mainly indicates non-fermenters. Example is Pseudomonas aeruginosa. Let us discuss some clinical case scenario. A 24-year-old male presents to the outpatient department with 8 days of continuous high-grade fever, headache, generalized weakness, abdominal discomfort, and loss of appetite. He reports drinking untreated water from a local source 2 weeks ago. On examination, temperature is 39.5 degrees centigrade. Pulse 78 per minute indicates relative bradycardia, mild hepatosplenomegaly. The laboratory investigations showed growth of gram-negative bacilli, which was oxidase negative, motile bacteria, indole negative, urease negative, but citrate is positive. TSI showed K by A with H2S positive without any gas. So H2S positive means blackening of the butt, gas negative means no cracks or lifting of the agar. So diagnosis is enteric fever caused by Salmonella typhi. Learning points are K by A plus H2S positive plus urease negative plus motile bacteria strongly suggests Salmonella species. Second scenario is a 58-year-old male in the ICU developed fever and purulent respiratory secretions after 10 days of mechanical ventilation. Laboratory investigations showed gram-negative bacilli, which was oxidase positive, catalase positive, pigment production present, motile bacteria. TSI showed K by K. So diagnosis is ventilator-associated pneumonia caused by Pseudomonas aeruginosa. Learning points are K by K reaction on the TSI test tube with oxidase positive indicates the organism is Pseudomonas species. Let us discuss some precautions during result interpretation. So first, use a pure bacterial culture. Always inoculate TSI agar with a pure isolated colony to avoid mixed reactions. Second, inoculate correctly. Stab the butt once with the straight wear loop and then streak the slant surface. Avoid multiple stabs which may give erroneous results. Third, do not over inoculate. A heavy inoculum can alter the pH and affect the interpretation. Fourth point is incubate at 35 to 37 degrees centigrade for 18 to 24 hours. Examine the butt carefully. Blackening due to H2S production usually indicates an acidic butt even if the yellow color is not marked. Lead acetate is most sensitive indicator for detection of H2S production. Sixth, check for gas production. Look for cracks, bubbles or lifting of the agar. Points to remember during interpretation of TSI test. If TSI test shows acidic slant and no change in butt, this could result from inoculation of gram-positive organisms that can use lactose aerobically. Second, failure of microbiologist to stab the butt. Recheck the growth on slant by doing gram stain and catalase test. Do not use the word gas to denote H2S production. Do not interpret the result without stabbing the butt. Disadvantages of TSI test. Acid production from fermentation of sucrose may inhibit the blackening of iron indicator. TSI agar should be used in conjunction with a urease test to eliminate the Proteus species and Salmonella species. Let us discuss some applied aspect of TSI test. Rapid identification of enteric pathogens. TSI helps to differentiate bacteria based on glucose, lactose, sucrose fermentation and gas production in H2S production. This provides an early presumptive identification before confirmatory biochemical test. Second, screening of stool pathogens. In cases of acute diarrhea, dysentery, food poisoning, TSI assists in screening organisms such as Salmonella enterica, Shigella dysentery. Examples are Salmonella mainly produces K by A with H2S positive, Shigella mainly K by A with H2S negative. Third, identification of urinary tract pathogens. TSI reactions help to differentiate common uropathogens like Escherichia coli, A by A with gas, Proteus mirabilis, K by A with H2S, Klebsiella pneumoniae, A by A with gas. Investigation of wound and soft tissue infections. TSI assists in differentiating gram-negative isolates recovered from wounds. Examples are Proteus mirabilis, Morganella morgani, Aeromonas hydrophila. Detection of hydrogen sulfide production, blackening of the medium indicates H2S production, helping to identify the organisms such as Salmonella enterica, Proteus mirabilis, Citrobacter fruindi. Differentiation of lactose fermenters and non-lactose fermenters. This is particularly useful when colonies are isolated from stool culture, urine culture, blood culture. Examples, lactose fermenters mainly showed A by A reaction, non-lactose fermenters K by A reactions. TSI is not a definitive identification test, it is a screening and differentiation test that guides the microbiologist towards the correct organism and subsequent confirmatory test. Please like, share and subscribe for more microbiology sessions. Thank you.